Today I want to talk about rethinking the traditional Western blot. So for today's talk, one second, there we go. For today's talk, I want to talk about the Incell Western assay. I want to go over what the assay is, how the assay benefits you in the lab, and how you can use the Incell Western assay for various application and experimental questions. I then want to touch on some things you should consider when you start optimizing the Incell Western assay from a traditional Western blot, and then go through quickly a brief example of how you can use this in research. So what is the Incell Western assay? The Incell Western assay is a high-throughput approach to simultaneously detect and quantify two separate proteins directly within a cell. By utilizing an Odyssey imager, you have two solid-state infrared lasers. These infrared lasers give you high sensitivity and low background for this assay. So let's go through an example workflow of the Incell Western assay. The assay begins with a microtiter plate, and for this assay you can use anything from a 6-well plate all the way up to a 1536-well plate. You seed your cells of interest, and your cells can be adherent or suspension cells. So you seed your cells and allow them to grow to the desired confluency. Once your cells are at that desired confluency, you go ahead and do your incubation. If you're interested in signal transduction, perhaps you add a ligand. Perhaps you vary the concentration of that ligand from well to well. Perhaps you increase or decrease the time of incubation from well to well, or add an inhibitor. If you're doing siRNA screening, perhaps you screen from well to well different siRNA and look at a target of interest. Once you're done with the incubation, you go ahead and remove the media, fix, and permealize the cells. Once the cells are permealized, you go ahead and you add a primary antibody specific for your target of interest. You then add a secondary antibody to detect your primary. Now, what makes these secondaries unique is that they're conjugated to an IR dye. These IR dyes will fluoresce in the infrared wavelengths and allow for detection on an Odyssey imager. So once you're done with the secondary incubation, you go ahead and take the entire plate itself and place it on either an Odyssey Classic, an Odyssey Assay, or an Odyssey CLX, and scan the plate. Now, let's compare a workflow for a traditional Western blot to the Incell Western assay. If you do these types of assays with a traditional Western blot, your in-cell culture, you do your incubations, remove media, and then lyse the cells. You then transfer that lysate to Eppendorf tubes and perhaps fractionate. Once your samples are ready, you then run them into SDS-page gels, transfer the protein to a membrane, and then you start doing your blocking and antibody incubation steps. With a traditional Western blot, these are a lot of steps that can be very time-consuming for you in the lab. So with the Incell Western assay, when you transition to it, once you're done with that incubation, you remove the media, fix, permealize your cells, and then go straight into antibody incubations. With the Incell Western assay, we have less steps involved with the protocol, saving you time in the lab and saving you time in the experiment. Now, one key difference with the Incell Western assay to a traditional Western blot is with the Incell Western assay, we're looking at cells in a native state. Because of this, when we choose a primary antibody, we want to ensure that primary antibody can recognize native state epitopes. Now, in addition to the time-saving elements in the protocol, we'll also generate data very quickly. If you perform this assay in a 384-well plate, that's 384 data points you can get in a single day. To get that same amount of data with a traditional Western blot, you would have to run roughly 43 10-well gels, assuming you're using one well for a ladder. So we save time in our workflow, we generate a lot of data quickly. In addition to this, the quality of that data has been increased. Here we're comparing an experiment done with a traditional Western blot to an Incell Western. The specifics of the experiment aren't important here, but what the group looked at were various mutants listed on the x-axis. On the y-axis, they're quantifying the levels of phosphorylation of a target protein. Now, the experimental question they were asking was, how do these different mutants differ in levels of phosphorylation of this target protein compared to controls? Now, the first thing they noticed when they compared the two experiments with a Western blot to the Incell Western assay is if you saw high levels of phosphorylation for a mutant with a Western blot, you still saw high levels of phosphorylation with an Incell Western. The biggest difference they found between the two assays was the reproducibility of the Incell Western assay was much greater than a traditional Western blot. So when you look at these two graphs, when you look at the top graph, which is our Incell Western data, you'll see the error bars are much tighter from replicate to replicate, mutant to mutant. The reproducibility has increased with the plate-based assay because we're no longer lysing cells, running SDS-PH gels, or transferring that protein to a membrane. Because we've taken all of these steps out of the equation, our reproducibility has gone up. So for this group, when they did the experiment with a Western blot, they could only find 4 mutants that had significantly higher levels of phosphorylation compared to controls. Same samples, same experiment done with the Incell Western assay, they now found 15 mutants that had significantly higher levels of phosphorylation over controls. So we save time in the workflow, we generate data rapidly, and the quality of that data has been increased. So let's walk through an example looking at protein phosphorylation. So in this example, we seed our cells of interest, and these cells express a receptor, let's say the EGF receptor. Once those cells are at the desired confluency, we go ahead and add our ligand, such as EGF. Once we've added our ligand, we'll have phosphorylation of downstream targets, such as ERK. Once we're done with the incubation step, we remove media, fix, and permealize those cells. Once the cell has been permealized, we can add a primary antibody specific for our phospho-ERK. We can then add a secondary antibody to detect our primary. Again, what makes these secondary antibodies unique is they contain and are conjugated to IR dyes. These IR dyes will fluoresce in the infrared wavelengths, allowing for high sensitivity detection on an Odyssey imager. Now in this example so far, I'm looking at just phospho-ERK. The Odyssey imager has 2 laser channels, so we'll look at our phospho-ERK in one laser channel. But what do we use the second laser channel for? Well, we would recommend that you normalize in the second laser channel. If you have changes from cell numbers from well to well, we need to normalize for that. Now there are a number of options you can normalize to. In my example, we're looking at phospho-ERK, so perhaps we want to look at total ERK in the second laser channel. If you don't have a total protein antibody, you can use something like GAPDH or actin, or another housekeeping protein. The downfalls or the disadvantages to doing a total protein or housekeeping protein approach is you have to use primary and secondary antibodies. These can be very costly. Additionally, you need to optimize the concentrations of each of these for your Incell Western assay, which can be very time-consuming. So many groups, rather than doing total or housekeeping proteins, will just do a nonspecific cell stain looking at cell number. Now there are a number of options out there when you just want to stain against cell number. Typically you might see DRAC5 or Sapphire 700 stains. DRAC5 is a nonspecific DNA stain. Sapphire 700 stain is a nonspecific nuclear and cytoplasm stain. Now if we took these 2 stains and plotted them against cell density in a plate-based assay like the Incell Western assay, what you'll see is for both DRAC5 or Sapphire 700 alone, their linear range for normalization is very limited. So the bottom trace is Sapphire 700 with circled data points. And we can see over this range of cell densities, it's not very linear. The middle trace with square rectangle data points is DRAC5 stain alone. And we could see it only works best at low cell densities. So typically researchers get around this issue by optimizing each stain separately and then combining the 2 to have a linear range for normalization over all the cell densities. Now rather than using both DRAC5 and 700 stain, Lycor offers CellTag 700 stain. It's a cell stain that we've optimized for use in the Incell Western assay that is a single stain that works both at the low and high cell densities. And if you look at the stain and plot it against cell density for an Incell Western assay, you can see you get a nice linear curve for normalization no matter which cell density you use. So once you've chosen your target of interest and you've chosen a normalization technique, we can go ahead and scan our plates and look at the final images. Here we're looking at a 96-well plate. Our red signal, our 700 channel, is looking at total ERK. Our 800 signal, or what's shown in green, is our phospho-ERK. The far left is a negative control background, and we can see we don't have any signals. And as we increase, or as we move from left to right, we increase the concentration of our ligand, EGF. As we increase the concentration of our ligand, we can see a correlating or a dose-response increase in our phospho-ERK. Now, using the Image Studio software associated with an Odyssey, you can easily put grids on top of this image. You can customize them to your particular plate and export the data to a graphing program of your choice. And in this example, since we're looking at a ligand, we can determine what is the EC50 for my ligand for increasing phosphorylation of ERK. Now, when you move from, so there are a number of other applications you can use the Incell Western for. These include gene expression, viral titer, apoptosis, RNAi screening, cell proliferation, and even protein trafficking. Now, I don't have time today to go through each of these. I encourage you, though, if you want more information about the Incell Western assay, that you visit Licore.com/ICW. When you visit our website, we have a lot of information about the Incell Western assay. We have protocols, publications, technical resources, and we go through each of these different applications and give you more specific examples of how you can use this in the lab for your research. Now, when you move from a traditional Western blot to an Incell Western, there's a few things you should think about. The first is, how do I want to fix and permealize my cells? Typically, you want to choose a fixing and a permealization agent that will work best with your antigen or your target of interest, as well as with that primary antibody. Most commonly, we see researchers use either a formaldehyde-based fixing agent or methanol. In addition to the fixing and permealization conditions, you also want to choose a primary antibody that will work well with the Incell Western assay. So the first question to ask is, does my primary specifically bind and recognize a single band? You want to test this by running a traditional Western blot, using the IR dye secondary antibodies, and examining that Western blot image on an Odyssey system. If you perform the assay and you see that your primary antibody specifically recognizes a single band, then you're ready to move into the Incell Western assay. If your primary antibody recognizes a number of non-specific bands, then you'll need to optimize your blocking conditions or perhaps even the primary itself before moving into the Incell Western assay. In addition to this, you want to ensure your primary antibody can recognize native state protein. Typically, you can determine this by looking at the antibody data sheet, and if that primary antibody has been validated for immunohistochemistry, then typically you can then go ahead and use that in the Incell Western assay. In addition to this, you'll want to use secondary antibodies that have been optimized for your Odyssey imager and the Incell Western assay. Lycore offers IR dye 680RD and IR dye 800CW, which have been optimized to work with an Odyssey imager for the Incell Western assay. In addition to this, you'll want to ensure your secondary antibodies are highly cross-absorbed to decrease any non-specific secondary binding to a primary. Now, when you go through these different optimization considerations, it's important to use a ZFactor analysis when determining which condition or which primary concentration to use. A ZFactor analysis will compare both positive and negative control replicates. It will compare the reproducibility from replicate to replicate, and it will also compare the separation between your positive control signal and your negative control signal. Now, utilizing the Image Studio software with an Odyssey, you can easily determine what is the ZFactor analysis for a positive and negative controlled data set. Now, let's go through a quick example of how you can utilize the Incell Western assay in research. And for this example, I'm going to talk about the Wnt beta-catenin pathway. Now, before I get into the actual details of the experiment, let's first remind ourselves of this pathway. So beta-catenin, just going through this little schematic here, is transcribed from DNA to mRNA and then translated into protein. Now, in a normal cell, once that beta-catenin has been translated, it's quickly phosphorylated and then degraded. If we add the ligand Wnt, it will block that phosphorylation and then cause an increase in concentration of beta-catenin inside the cell. Once the concentration of beta-catenin has increased, you'll see a correlating change in transcription inside the cell. This pathway has key roles in cancer biology and traditionally has been studied by either Western blotting or overexpressing beta-catenin in an artificial cell system. The issues with these approaches is, well, with a Western blot, you can't generate data quickly, and your reproducibility isn't high. By overexpressing beta-catenin in an artificial system, we can't ask true questions about how beta-catenin would normally act at a basal level inside a cell. So what Hanush et al. Did at Genentech in 2008 was to utilize the Incell Western assay to study this pathway. They chose cell lines that normally express beta-catenin at normal cell levels. They seeded these cells in 384-well plates, allowed them to grow to desired confluency, and then add their ligand of interest. In this case, they added Wnt3a. They would incubate the cells with their ligand, fix, permealize, and then they would add primary antibody specific for beta-catenin. To detect their beta-catenin, they would then add a secondary antibody with IR-dye 800 and detect this in the 800 channel of an Odyssey system. To normalize from well to well, they used DRAC5 as a DNA stain. In their first control experiments, when they looked at cells with or without ligands, they could readily see that when they would add ligand Wnt3a, they would see a correlating increase in beta-catenin expression and content inside these cells because Wnt3a would be blocking phosphorylation of beta-catenin and inhibiting degradation. They could then normalize to their DNA stain, export the data to a graphing program of their choice, and readily see and quantify this response. Now the benefit to an Incell Western assay utilizing a 384-well plate is you can do a dose-response curve in a single plate in replicates and readily determine what is the EC50 for my ligand for this increase in beta-catenin expression or content inside a cell. Now, by utilizing the Incell Western assay, this group could ask very specific, detailed questions about this pathway. I don't have time to go through all of the different pathways and questions they asked, but to highlight a few, their first question, or one of their first questions, was, well, if we would add ligand, we would see an increase in beta-catenin content inside a cell. Was this increase in concentration due to an increase in transcription of mRNA, or an increase in translation from mRNA already existing inside the cell? To test this, they would add Wnt3a alone, or Wnt3a with cyclohexamide or actinomycin D. Actinomycin D inhibits transcription of new mRNA from DNA. Cyclohexamide will block translation of new protein from existing mRNA. Now, when they would look at these cells with ligand and actinomycin D blocking transcription, they found that this had no effect on increases in beta-catenin concentration inside a cell, suggesting that when you would see an increase in beta-catenin concentration, it's not dependent upon transcription. When they would block translation with cyclohexamide, they would see that they could block this increase in beta-catenin concentration, suggesting that when they would add ligand and see an increase in concentration of beta-catenin, that's dependent upon existing mRNA pools and not dependent upon transcription. In addition to asking detailed questions like this, they could ask new questions about this pathway. So what they did was a temporal analysis. They would take a 384-well plate, seed their cells of interest, and vary the concentration from well to well across this plate. For each of these different ligand concentrations, they would also vary the time of incubation and quantify how does the concentration of beta-catenin inside these cells change dependent upon 1, time, and 2, ligand concentration. By looking at the dose-response curves for these different time points, they could readily determine what is the EC50 for Wnt3a with these different times of incubations. And they could ask new questions such as, how does the rate of beta-catenin accumulation change dependent upon Wnt3a concentrations? For this group in particular, by utilizing the Incell Western assay for the Wnt beta-catenin pathway, they found it gave a very cost-effective way to examine the pathway. Since this is a 384-well plate, you don't need to use large volumes of reagents. You don't have to use a lot of costly ligand to do these studies. You can do them in the microtiter assay and generate a lot of data quickly. By doing experiments like the temporal analysis, they could get unprecedented level of detail of this pathway. So for this group in particular, they found that they could then move on and screen or look at validation of modulators to the system, or perhaps extending it to different cell lines or different cell types and asking how does this pathway differ in a different cell system. So to summarize, the Incell Western assay gives researchers the ability to, one, save time in your experiments, save on your reagents, and ultimately the data you generate be of higher quality. Again, with the limited time today, I don't have the time to go through all of the different applications and uses, so I encourage you, if you want more information about the assay, to visit lightcore.com/ICW. I also encourage you to visit lightcore.com/custom services. If you visit our custom services website, you have the ability to get in direct contact with a Lightcore scientist and get their opinion of how to optimize this assay for your particular experimental question. In addition to this, you can send Lightcore your cells, your antibody of interest, and our custom services program will go ahead, optimize the protocol for you, and then give to you a final protocol that will address your scientific question, and you can follow that and then use the Incell Western in your experiments.